Hello, dear colleagues. My name is Petar Jovanovic, and I'm a PhD student at Ben Gurion University of the Negev, and my research has been conducted in the GLAD Research Center, which is a part of Agricultural Research Organization in Israel. And today, I will be talking about night brought to light, daily calcium homeostasis in tomatoes, fruit during stress. As it's very well known, transport of calcium to tomatoes, to generally fruits and tomato fruits, is very much dependent on many factors. It's a multifactorial process, but processes as salinity or drought can influence this process of a movement of calcium uptake and movement of calcium from the soil through the plants and into plant fruits. And in the cases where salinity, drought, and some other factors influence or stop, or in any case diminish the transport of calcium, this can lead to different physiological disorders. One of them is blossom end rot, which you can see on the picture here on your right-hand side. This is a dark spot which develops on the bottom of tomato fruits. It's a multifactorial physiological disorder, but calcium is one of the main factors that leads to its development. Moreover, similar physiological disorders can show up in peppers, in apples, in some other fleshy fruits, and this can lead to a very big impact in to producers. This kind of fruits are very hard to sell, and this can lead to very big economical damages to the fruit producers. Moreover, nighttime transpiration. Nighttime transpiration has been known to happen for a very long time, but its role in physiology of the plants is not that well known. It's also a multifactorial process, but the specific roles of this process are not that well known, but it has been shown to happen. We have conducted our measurements, and between 8 to 10% of all evapotranspiration that happens in tomatoes happens in the night specifically. This leads us to our hypothesis of our work. In stressful environments, like the picture of the Negev that you can see on your left-hand side, in this kind of dry and hot environments, will more favorable conditions during the night lead to better nutrient acquisition? Moreover, improving plant response to different kinds of stressors. To try to answer our hypothesis, we formulated an aim. An aim was to establish a function of nighttime transpiration and stress in calcium delivery to tomatoes. We conducted several experiments in different environments, and I will say something about it on the next slide. But in all of those experiments, we manipulated the environment, manipulated the evapotranspiration that happens by manipulating humidity, increasing and decreasing humidity, and increasing temperature, increasing the wind speed. Moreover, we use strontium as a tracer for calcium. Strontium has been known for years to be used as a good tracer for calcium, but this is something that it's not actually that well established, because strontium sequestration happens in different physiological conditions, in different environments. So we need to show, is strontium a good tracer for strontium? We conducted several experiments, long-term and short-term experiments. As you can see on your left-hand side, you can see short-term experiments conducted in hydroponics between 2 to 20 hours. That's how long they were. And in those control environments, we manipulated the environment, wind speed, humidity, and temperature. While on your right-hand side, you can see how long-term experiments looked like. They were conducted in a length between 26 to 40 days, and they were conducted in net houses and greenhouses in perlite. In all of them, strontium was used as a tracer for calcium. Now I will present our results. As I said, we first need to establish, is strontium a good tracer for calcium? Our results in a long-term experiments show that long-term allocation of calcium and strontium is very much comparable, and that strontium compensates the missing calcium, and that both calcium and strontium accumulate at the same rate. And on your right-hand side, you can see panel showing accumulation of strontium and calcium in the shoot. And on the left-hand side, you can see accumulation of strontium and calcium in the fruit. Something that we also observed and something that we report for the first time is that strontium has strontium is mitigating blossom and rot symptoms. As you can see on the left-hand side on the bottom panel, you can see the percent of blossom and rot in the group that received calcium. Blossom and rot was greatly diminished, and it diminished at the same rate as the addition of calcium. And on the picture on your right-hand side, you can see the actual fruits. Fruits that received calcium, fruits that received strontium, and fruits that didn't receive any of those elements. The next step was to also establish connection between strontium and calcium, but this time in short-term experiments, to establish uptake rate and pathway of strontium and calcium, and we conducted different short-term experiments in hydroponics, as the picture you can see on your right-hand side. On the panels, which you can see, these the first upper panel, you can see the time frame. It's night and day. These are uptake rates of strontium and calcium in night and day. And we can see that strontium and calcium uptake are comparable in hydroponic solutions. More hydroponic conditions. Moreover, and on the bottom panel, you can see the results of our blockers experiment. We use lanthanum and gadolinium, which are specific calcium blockers, to show if they will decrease the uptake of calcium at the same rate as strontium. If they will increase the uptake of strontium in the same rate as calcium. And this is what we show. On the panel, you can see that strontium and calcium uptake have been decreased at similar rates, showing that calcium and strontium share the uptake. Something that we also show, and which is actually very much known from the literature, is that strontium has some very negative effects. You can see here on all these three panels, on your left-hand side, you can see a panel of stomatoconductants on the up, and on down you can see the panel showing the net photosynthesis, and we can see that in group that received strontium, there is a significant decrease in stomatoconductants and net photosynthesis, showing the negative effect of strontium application in long-term experiments. These are results showing in the long-term experiments between 26 and 40 days. On the right-hand side, on the panels on the right-hand side, you can see a biomass, plant biomass, and you can see that in the group that receives strontium, biomass is significantly decreased, showing again that strontium has some negative effects in long-term experiments. Moreover, we conducted experiments in day and night. As we said, our hypothesis depends on premise that night can provide some benefits when it comes to day. When the stress is lower during the night, that this will provide some sort of a benefits to the plant. And we conducted experiments in which we evaluated the uptake of strontium and calcium in night and day. And these are the panels showing the strontium uptake for shoot and fruit. And on the left-hand side, you can see the shoot, and that most of the strontium is uptaken during the day, while only half as much of strontium is uptaken during the night. On the right-hand side, you can see uptake in the fruits. And this is very interesting, that most of the 99% of the strontium is uptaken to the fruits, delivered to the fruits during the night, showing that night is the critical time for tomato fruit calcium delivery. And this is the meat of our presentation that we're going to talk about today. And this is results showing delivery of strontium, calcium, to the fruits during stress, to tomato fruits during stress. So we conducted different experiments in control environments, in the net houses, in the growing rooms mainly, where we subjected plants to strong wind, to humid or dry condition to hot or cold conditions. And on the left-hand side, you can see a graph showing our one representative experiment, while on the right-hand side, you can see the graph showing all 5 experiments collated together. And something that we show is that environments which promote evapotranspiration will decrease the delivery of calcium to the fruits, meaning that in the environments which promote low evapotranspiration, transport of strontium to the fruits will be increased. Calcium delivery to the fruits is very much dependent on evapotranspiration, and low transpiration conditions increase the portion of calcium delivered to the fruits, meaning that in stressful environments which promote evapotranspiration, like hot and dry environments, most of the strontium calcium will not be transported to the fruits, but this will happen in the environments which evapotranspiration is low. And this is something, just the last slide, a take-home message for you all today, is that strontium can partially replace calcium in tomato, strongly mitigated blossom and growth. Strontium is a good short-term tracer for calcium in tomatoes, while in the long term can lead to negative effects in plants. Calcium allocation to tomato fruits is mainly done during the night. And the last point is calcium allocation to tomato fruits is strongly related to environmental conditions, mainly that in conditions that decrease evapotranspiration, strontium allocation, calcium allocation to the fruits is being increased. Thank you very much, and that will be all for today. Thank you.